Hello everyone, I'm here to present our recent work on using a feature selection pipeline to identify target proteins predictive of drug toxicity. Toxicity assessment has always been a critical part of drug development research because toxicity is a major cause of attrition, accounting for around 30% of clinical trial failures. There are experimental approaches to test toxicity, but they are expensive and cost a lot of time and labor. That is why we need computational tools to predict whether a compound can cause certain toxic events based on its chemical structure. A commonly used method in the field is QSAR, which starts by quantifying chemical structure into a feature set, then relate the feature set to toxicity outcome using classification models. However, toxicologists do not like QSAR very much. To start with, the accuracy is not very satisfying. And also because of its nature, QSAR models cannot point us to the proteins or pathways which may help explain cellular mechanism of toxicity. To address the limitation, we came up with a two-step strategy. In the first step, we built QSAR models to predict binding of compounds to their target proteins. Because even though QSAR models often fail to predict toxicity, they have quite decent performance when used to predict compound-target interactions. And we can do this for hundreds of target proteins we have data for to get the target binding profile of each compound. Then relate the target binding profile to toxicity in step 2. Because previous studies have found drugs of alike toxicity outcomes often share targets. But this strategy presents us with a new challenge. As you can see, in both steps, the amount of features we use to describe compounds is pretty close to the limited number of samples we have, and that could lead to overfitting of the models. To prevent overfitting, we develop and implement a feature selection pipeline named TargetTox to identify features that are predictive of the outcomes first, then only use the predictive features for classification in step 1 and 2. A key advantage of TargetTox is it can detect and preserve feature interactions during its selection process. And interactions can be quite prevalent among our chemical or target features. So in step 1, we implement TargetTox to identify molecular descriptors that are predictive of binding to 569 target proteins. As you can see from the scatter plot, where each point represents model performance for a single target, that using only the predictive descriptors identified by TargetTox, we are able to achieve the same level of performance as using all descriptors for classification. And that means the selection process of TargetTox works. We also compare TargetTox to a common selection method, L1 regularization. And as you can see here, TargetTox outperforms L1 by a large margin, with a much smaller set of predictive features, and that makes our model much easier to interpret. Of all the 569 target proteins we studied, around 2/3 of them have an AURC greater than 0.85. And these target proteins cover a variety of functional classes. We also found that if two targets are both catalytic receptors or GPCRs, they are more likely to have similar sets of predictive descriptors identified by TargetTox. And that is what we would expect for targets of similar functions. In step 2, we implemented TargetTox to identify targets predictive of 815 adverse events. And again, we compare the performance by predicted targets to performance by all targets. We found the points are mostly aligned along the diagonal line, like in step 1, but with a much larger variance. And the same thing happened when we compared predicted targets to conventional structure-based QSAR models. That means through TargetTox, we only achieve the same level of performance as conventional QSAR models, with no significant improvement. But we still think TargetTox can beat QSAR in terms of interpretability. Because if we go back to the predicted targets, we can see if they can help us to explain cellular mechanism of toxicity. For instance, when we mapped 15 adversaries of interest to their tissues of toxicity, we found the predicted targets identified by target tox are more likely to be differentially expressed in the matched tissues. And that suggests these targets can have important functions in the tissues. And if these functions are disrupted by drugs, then that could lead to abnormal activities in the tissues. To better understand the important functions of predicted targets, we performed functional enrichment analysis. And we were able to recapture some key toxicity-related functions. For instance, we found that targets predictive of ventricular dysfunction are enriched for Notch signaling pathway, which is required for ventricular development. They are also enriched for cellular response to epinephrine, which is often used in the treatment of ventricular fibrillation. So both these two function terms can play an important part in ventricular dysfunction. We also validated our predict targets against known disease markers. As you can see, 2 adverse events in skin and liver are significantly enriched for the disease markers of the matched categories. And there are 4 additional adverse events have odds ratios greater than 2. In addition to recapturing known disease markers, target tox can also identify new potential markers of toxicity. For instance, this dehydrogenase HSD11B1 is found to be predictive of acute liver injury. We found this gene is highly expressed in the liver, and a recent study revealed that inhibitors of HSD11B1 can promote fibrosis in mouse model of liver injury. So potentially, this dehydrogenase can act as a new marker for liver toxicity. So to give a brief summary of our project, we came up with a new strategy to better explain toxicity by connecting chemical structure to toxicity outcome through target binding profiles. We presented a feature selection pipeline named TargetTox to identify predictive targets. We demonstrated that these predictive targets can help explain toxicity by showing their specific expression in the tissues of toxicity, their enrichment of toxicity-related functions, their connections to diseases as markers, and their potential to be new markers for toxicity. And that concludes my presentation. Thank you all for your attention.